Hello, my name is Alexis, and we are here to inform you all about nicotine dependence. We are also here to advocate for a genetic test we have created to discover if you have specific genes that will make you more susceptible to nicotine dependence. Smoking is a significant issue in America. The CDC even reports that 34 million adults in the United States smoke cigarettes. Because nicotine is addictive, it can lead to many harmful consequences. The CDC also reports that cigarette smoking alone kills more than 480,000 Americans each year, and 60 million Americans are living with at least one smoking-related severe disease. These staggering numbers could be the result of one's susceptibility to nicotine dependence upon smoking cigarettes. Nicotine dependence is one's habitual and chronic tendency to consume nicotine as if it is a necessity, typically leading to that person's inability to stop using nicotine. Research has demonstrated that genetics significantly influences someone's susceptibility to nicotine dependence, proving to be about 40% to 60%. Some estimates even claim that genetic backgrounds account for 80%, but it seems that the most accurate contribution is around 67%. NACH receptor proteins, specifically its alpha 4 and beta 2 subunits, which are the most abundant of the NACH subunits, are essential components of the rewarding effects of nicotine. The National Library of Medicine says that these proteins are located at the skeletal neuromuscular junction where they mediate neuromuscular transmission, and neuronal receptors, which are found throughout the peripheral and central nervous system where they are involved in fast synaptic transmission, and that the NACHRs have been demonstrated to be involved in cognitive processes such as learning and memory and control of movement in normal subjects. It makes sense that the variation changes the structure of the NACHR's proteins because they alter human brains and play a significant role in nicotine addiction.
The SNP chip test we have designed will include 10 different genetic variations. Two genetic variations we will briefly discuss are the RS1051730 and RS10485057. First, the RS1051730 is strongly associated with lung cancer, but researchers believe there to be a direct correlation between lung cancer and nicotine addiction within this SNP. Having a double copy of the T allele is associated with a 1.8 times higher chance of developing lung cancer. Those who have either one or two copies of the T allele are more likely to smoke than those with the homozygous C genotype, which as a consequence would increase the risk for lung cancer. This establishes a direct link between the likelihood of developing lung cancer to that of first having a nicotine addiction. There aren't any specific changes in the gene listed on the SNPpedia website, but based on the increased use of nicotine and alcohol abuse of people with this SNP, I could hypothesize that it affects the speed at which new substances are processed in the body somehow. If it takes longer for the nicotine or alcohol to have its effects, you're more likely to consume more of the product. The RS10485057 SNP would also be an excellent SNP for this test. The only nicotine addiction research listed on the SNPpedia website is for Caucasian men. However, studies that show a link to depression in African American populations and an effect on the OPMR1 gene in the Spanish population are also shown. This is a genetic variation in the mu1 OPMR1 gene, which is listed as an opioid receptor. The OPMR1 gene provides instructions for making a protein called the mu opioid receptor, which is responsible for feelings of pleasure and pain control. Changes in this protein could have effects on how someone feels pain or pleasure and reacts to substances that have similar effects.
There are many benefits to completing the test we have designed. If you take the test and it comes back positive that you have the specific genes we believe are linked to increased risk of nicotine dependence, then you know once you smoke, you'll have a very difficult time to stop. An experiment was conducted by the Virginia Institute of Psychiatric and Behavioral Genetics helped further how reliable our test is. They concluded that out of all of many different gene expressions, that RS1048507 had the strongest correlation to nicotine dependency. Brigham and Women's Hospital studied the relationship between the SNP RS1051730 and nicotine dependence. They concluded that in a room full of currently smoking subjects, the A allele of RS1051730 was associated with an increased Fagerstrom test for nicotine dependence score. This evidence shows that if you take our test and you show that you have this gene, more specifically the A allele, you are predetermined to be more susceptible to the nicotine dependence. These two studies shown on the screen express how prominent the gene expressions we put in our test have been proven to have a strong correlation of nicotine dependence. If you take our test, you can have faith that your results will be accurate. As a startup company, we want to be very upfront with the benefits and the limitations that come with taking our test. One limitation is that there are more than one genetic variation that has been said to lead to nicotine dependency. This being said, it is hard to target a specific gene and say if you are going to be predisposed to the dependency or not. A genome-wide study of more than 38,600 former and current smokers concluded a DNA variant located in the DNMT3B is identified and contributing to nicotine dependence. This shows that there's more than one gene expression that leads to this dependency. Although we test for genes that have correlation to the nicotine dependency, our test does not account for every gene linked to the dependency. There's also many other factors that contribute to nicotine addiction that our test cannot account for, including social influences, environmental influences, mood disorders, etc. While the genetic alterations we test for have been shown to have a strong correlation with the nicotine dependence, whether or not the SNPs themselves are the cause is unclear, or whether we can associate the nicotine dependence with other disorders caused by the SNPs, such as depression or anxiety.
Lastly, we wanted to discuss further other warnings and ethical considerations that come with a new test we are proposing. Allison Wright from Thomas School of Medicine found that testing for genetic susceptibility to nicotine dependence could actually encourage smoking behaviors. If an individual learns that they are not susceptible to nicotine dependence, they may take this as a green light to begin or continue smoking. Or those who find out they do have a genetic variation, they may feel hopeless when it comes to avoiding nicotine dependence. Discrimination upon finding out about one's susceptibility to nicotine dependence is also possible. Many physicians have expressed concern about discrimination with insurance or in the workplace after finding out their predisposition to nicotine dependence. The GINA Act, passed in 2008, makes it illegal for health insurance providers to raise premiums or deny coverage to individuals based on genetic test results. However, this does not apply to disability insurance, long-term care insurance, or life insurance. Thank you all for listening. I hope we were able to inform you about our new genetic test we've created for nicotine dependency.